For today's webinar, we have Dr. Sam Kwiatkowski from the Harris County, Texas Institute of Forensic Sciences presenting a DNA barcoding strategy for blow and flesh flies encountered during Medicolegal casework. Without further delay, here is Dr. Kwiatkowski.
The authors do not have any financial interests or relationships to disclose. Hello, my name is Sam Kwiatkowski, and I'm a researcher with the Harris County Institute of Forensic Sciences. Today, I'm going to talk about a method we are building to assist forensic entomology practices for our agency. Our primary use for entomology is to gain insight into the postmortem interval for forensic casework. Insects have species-specific patterns in their biological development and community succession, which may be used to estimate their time of colonization, which may then be used to approximate the postmortem interval. Before an estimate can be made for time of colonization, the taxonomic identification must be made for specimens. This is typically carried out by examining morphological traits. However, this has disadvantages. First, the specimens most relevant for estimating time of colonization are the immature types, the larva, pupa, and sometimes pupal exuvia collected from or around the body. Many species cannot be identified to the species level from these stages. Second, examination may be ineffective when specimens are damaged or poorly preserved. Third, it is possible to rear live specimens into adults, which may then be examined for morphologies. But this is resource intensive and is not always successful. Specimens may not survive, or they may turn out to be female, which would not be helpful for some species. Finally, medical examiners and crime laboratory staff typically do not have the expertise necessary for examining morphologies. This sometimes requires skills for dissecting minute traits that are inherently difficult to study and not obvious to the untrained eye. Alternatively, taxonomic identification may be achieved through DNA barcoding. This is a molecular identification technique where DNA sequences from specimens are matched to references. This may be performed without specialized forensic entomology training, and it can be done using equipment and methods that are commonly available in forensic genetics laboratories. This process includes PCR amplification from a target locus, Sanger-based sequencing of the amplified fragment, and statistical analysis for comparing sequences to references. Today, I'm going to describe our method and the range of species that we have tested. I will also describe the process of analysis, which includes comparisons to published references, as well as sequences derived from the local populations here in Harris County, Texas. I will also describe experiments aimed at validating our implementation and quality control steps. These will include experiments describing the sensitivity of the assay, tolerance to challenging conditions that may be encountered, accuracy tests using known specimens, and a demonstration using unidentified casework specimens. An early step in our process was to build a list of species that are most relevant. Our list was informed by trends in recent casework and regional surveys. Our list consists of 15 blowfly species from genera Califera, Lucilia, Chrysomya, Cochleomya, Phormia, and Cynomya, as well as the flesh fly Lazocypha and the scuttlefly Medicolegal scalaris. All of these are considered to have great forensic interest not only in our area, but also across the United States. This list shows the frequency that these species have been observed in our casework. These rates include 8 species that appear in at least 7.6% of the time. Chrysomya ruffifacies was the most common of these and occurred in almost 38% of our casework. Medicolegal scalaris, Chrysomya megacephala, Cochleomya macellaria, and Lazocypha were observed in 17 to 29% of casework. Phormia regina and Lucilia eximia were observed in nearly 10%. Lucilia caprina was observed in 7.6%. Various other blowfly species have been observed in a smaller number of times. These include additional members of genera Califera and Lucilia, and also Cynomya cadaverina. The next step was to select a primer set for amplifying a barcoding fragment from each of our target species. The locus we targeted was the mitochondrial locus cytochrome oxidase, or CO1, which is commonly used for this purpose, including for forensically relevant insects. We looked to the literature for primer sets. We understood that it would be advantageous to find a single primer set capable of amplifying all of the species on our list. Unfortunately, this was hampered by their diversity. The literature does not clearly demonstrate a single primer set that would be sufficient. A common solution used in academic studies would be to apply additional primer sets to ensure the appropriate coverage. But this would be undesirable for our assay because we want to minimize the workflow as much as possible. Our intent will be to analyze unknown specimens, We would never know beforehand which primer set would be best for any particular specimen, so we would have to perform multiple reactions per specimen. Our solution was to test multiple primer sets from the literature until we found one that would work for all the species on our list. This additionally extended to all the PCR cycling conditions and reaction compositions, which should also be the same for all species. We tested many primer sets from the literature. Some of these targeted the 5' region of CO1 as used for the Barcode of Life database. Other primer sets targeted the 3' region as demonstrated for forensically relevant insects. We tested primer sets using verified specimens. These were adults that were provided to us by an outside expert and whose identities had been confirmed using morphologies. Each of the primer sets that we tested amplified from a majority of the species relevant to our assay. But there was only one primer set we found to be successful in amplifying from all of the species relevant to the assay. This produced the appropriate size band at 560 base pairs. In addition, we did not observe any additional reaction products which other primer sets produced for certain species. The primers corresponded to the literature designations C1J2495 and TL2N3014, which target the 3' region of CO1. The sequences were slightly modified from the literature to match consensus sequences for target species. We did not use degenerate primer sequences. The reaction and cycling conditions were standard for all species. We also did not require a touchdown PCR step or additional annealing steps at more permissive temperatures. This slide shows the processing steps that are carried out in our laboratory and estimates for the time required at each step. First, DNA is extracted using an insect-specific kit. Extraction includes time for lysis, an organic extraction, and purification using spin columns. Second, the extracts are quantified using a fluorometric assay. In this case, the assay will measure total DNA content rather than giving a specific measurement for mitochondrial DNA. Next, the PCR reactions are prepared. We use the Q5 high-fidelity enzyme in the master mix preparation. Then, the products are visualized by gel electrophoresis. After amplification is confirmed, we send the reactions to an external company for amplicon purification and Sanger sequencing. The entire in-house processing can be completed easily within 2 days if necessary. Bidirectional sequences are produced using our forward and reverse PCR primers. This slide shows sequencing output from sense and antisense reads. We perform a few quality control checks before moving forward. First, we look at the Phred scores, which are shown in color code. Any nucleotides that are highlighted in blue indicate scores that are at least 20. Any nucleotides not reaching this threshold are trimmed or changed to the letter N as an ambiguous call. Second, we look for the opposite primer sequences at the end of the read. These sequences are underlined in the image here. 8 to 15 base pairs are typically visible before the reads terminate. Finally, we check the total length of the reads. These will vary due to limitations in the method, but should be around 520 base pairs or greater. These checks support that we have sequenced the CO1 locus rather than a CO1-like sequence, which may be produced from nuclear-encoded duplications of CO1. CO1-like sequences will have additional polymorphisms and potentially insertions or deletions. Our assessment is meant to support that the product resulted from application using both forward and reverse primers, that the sequence does not contain any insertions or deletions, and that the results are not impacted by spurious amplification products. Once we have confirmed these assessments, the bidirectional reads are tiled to produce a sequence corresponding to the full barcoding fragment. The size is 514 base pairs after removing the primer sequences. We obtained full-length sequences for each of the verified specimens. These were next tested using phylogenetic analysis to determine whether the species may be distinguished using our CO1 fragment. We are using the online tool phylogeny.fr, which streamlines several programs necessary for this analysis. These include processes for sequencing alignment, maximum likelihood tree building, and tree visualization. The trees are midpoint rooted, which means that the sequences are divided at each node into 2 groups over several iterations. This process does not use outgroups, therefore the trees reflect differences in the sequence without consideration for evolutionary relationships. The tree building algorithm additionally comprises non-parametric bootstrapping for statistical support. Under this process, the algorithm is repeated for a user-defined number of replicates, with the data being resampled for each replicate. The number of replicates used in our analysis was 500. Published reference sequences were used for phylogenetic analysis. Sequences for representative specimens from the United States were selected if possible. There are some exceptions. Some species were not native to the United States, but were necessary for certain comparisons. Otherwise, suitable sequences were not always available for respective species. This slide shows an example tree produced using phylogeny.fr. We input published sequences from North America, as well as sequences produced in-house from verified adult specimens, which are labeled here in the tree as VER. We additionally included published sequences for closely related groups that were necessary for comparisons. The values shown in red represent the percentage of 500 bootstrap replicates that support each node. There is additionally a key in the bottom left corner that relates nucleotide changes to horizontal distance in the tree. The value represents the percentage of the overall sequence length. Here, this value is 0.2 or 20%, which corresponds to about 103 nucleotides. We observed several trends in our analysis that are consistent with the literature. Many species relevant to our assay were observed as monophyletic, that is, their sequences were isolated from all other species in the tree. In this analysis, these groups exhibited at least 91% bootstrap support. This suggests that these species may be easily distinguished. There were also several species which were not monophyletic. In the next few slides, I will discuss more specifically about some of these species. For many species, identification may be supported. For others, identification may be more difficult. This slide shows the portion of the tree comprising sequences for the flesh fly, Blazocypha pica. We often find these cases where the specimens present are dominated by flesh flies. These are typically scenes that occur indoors. Flesh flies typically can only be identified to the species level when examining adult males. In the context of death investigations, making identifications will not be possible unless we are capable of rearing specimens to adults and if males are recovered. In the past, when this has been successful, we have found that these scenes are dominated by Blazocypha pica, which is why this was the only flesh fly that we targeted. Being able to identify this species through DNA would greatly improve our ability to provide time of colonization assessments for these scenes. This is one example where we included sequences for species which were not targeting, but needed to include to improve our analysis. We included a sequence from Blazocypha cessator. This was the most similar non-Blazocypha pica sequence we could find. All Blazocypha pica sequences clustered independently of Blazocypha cessator. Another group that we considered was genus Cochleomya. There are 4 members: Cochleomya minima, Omnivorax, Macellaria, and Aldrichi. This analysis showed a monopoly for Cochleomya minima and Omnivorax. The former is isolated to the Caribbean. The latter is a livestock pest that has been eradicated from North America, but still inhabits South America and the Caribbean. In 2016, this species was reintroduced in Florida Keys, which caused eradication to be reinitiated. We do not consider Cochleomya omnivorax to be one of our main targets for forensic casework, but our assay could be used to detect them if they are reintroduced in our area. We additionally considered Cochleomya macellaria and Aldrichi. These could not be separated using CO1. However, they have very different geographic distributions. Macellaria is one of the most common species in our area, whereas Aldrichi is isolated to the Caribbean. This is an example where the dramatic differences in distribution are helpful for our analysis. Any specimens that cluster with this group are likely to be Macellaria rather than Aldrichi. The next interesting group was from genus Chrysomya. We are interested in 2 species. First, Chrysomya ruffifacies was found to be monophyletic. The other, Chrysomya megacephala, was not distinguished from relatives Chrysomya safranea and pacifica. However, this is another situation where the geographic distributions are helpful. Chrysomya megacephala is one of the most common species in our area, but the others are isolated to Southeast Asia and Australia. Therefore, specimens that cluster in this group are likely to be Chrysomya megacephala. The next species are Lucilia caprina and Serracata. These are closely related species for which we did observe reciprocal monopoly in our analysis, but still require caution. There are 3 haplotypes here: Lucilia serracata, Lucilia caprina, and hybrids that have characteristics of Lucilia caprina but harbor a Serracata-like haplotype. The major concern with these species is the possibility of hybridization. This is what permitted the Serracata-like haplotype to form in Lucilia caprina at some point in the past. Hybridization is also suspected among the modern species in areas where they overlap, which includes our region. Because we would not be able to distinguish between these hybrids, we expect to avoid making calls between these species. There was a similar situation between Lucilia eximia, mexicana, and Cerula viridis. These are related species that inhabit different areas of the country, but have overlapping boundaries. Lucilia cerula viridis inhabits mid to eastern regions of the country. Mexicana inhabits mid to western regions, and eximia inhabits regions along the Gulf Coast. Distinct haplotypes have been observed for regional populations of both Mexicana and eximia. Lucilia mexicana has a distinct haplotype found in Mexico and in the United States along the Pacific coast. Lucilia eximia has multiple distinct haplotypes that occur throughout Central and South America. Lucilia eximia and mexicana may also exhibit a Cerula viridis-like haplotype in regions where they overlap, which for all three species includes Texas. This is another group for which we expect we would not try to separate because of the overlapping distributions and suspected hybridization. The remaining species are groups that we consider relevant to our assay, but require additional development data before they can be used for time of colonization estimates. Sequences for Lucilia illustris and Caesar are identical, but illustris is a New World species, and Caesar is an Old World species, so these would be distinguishable when considering their distributions. Calurifera coloradensis and livida are closely related as well. Both are widespread here and appear to share haplotypes according to published references. Another concern for these groups, as well as for Megacilius scalaris, is that few sequences have been published for specimens in the United States. A future goal of ours would be to establish references from our area. This slide compares our expectations for successfully identifying species versus actual encounters in our casework. There are select members of genera Lucilia and Calurifera, which we may not be able to distinguish using this assay alone. We may also identify Megacilius scalaris and Lucilia illustris, but these require additional development data. However, there are 8 species which may be distinguished when combining sequencing results with known distributions. These include 5 species that appear in at least 10% of casework. Chrysomya revifaces was the most common species, occurring in almost 38% of our casework. Chrysomya megacephala, Cochleomya macellaria, and Blazocypha pentapyga were observed in about 17 to 24% of casework. Phormia virginia was observed in almost 10%. This suggests that our assay would be effective for the majority of species that we know are common in our casework. Next, I will describe a few selected studies from our validation. These are meant to elaborate on our implementation and describe additional quality control measures. These specifically will be sensitivity of the process, impact of challenging conditions, blinded mock casework experiments for accuracy, a study of local populations in Harris County, and finally, I will demonstrate analysis of unidentified specimens with statistics to support identifications. In terms of sensitivity, we were wanting to evaluate the impact of template amounts on the quality of gel electrophoresis and sequencing data. We needed to determine template amounts where we would expect the assay to fail. In this particular experiment, we observed a band at the appropriate size going all the way down to 0.25 nanograms. These inputs reflect total DNA content and is not specific to mitochondrial DNA. When we reviewed the sequencing data, we found poor quality sequences at the lowest template amounts. The best sequencing results were observed when we amplified at least 0.75 nanograms template. No difference in quality was observed for greater template amounts. These template amounts were easily obtainable in view of the sample types we expect to test in casework. Larvae and pupa will yield ample material from a small cutting. Adult legs also usually yield plenty of template. We routinely observe about 10 to 30 nanograms per leg. We tested a few reaction conditions that we expect to challenge our process. On this slide are results using degraded or inhibited reactions. These are standard types of studies that we include when evaluating any PCR chemistries. For PCR inhibition, we spiked reactions with increasing amounts of humic acid, which we found stifled any observable amplification. For degradation, templates were autoclaved over an increasing amount of time, and this also was greatly detrimental to amplification. We additionally tested specimens that had been in storage for a period of time. These specimens were dehydrated in alcohol and stored at room temperature with desiccants and vapor tape. Amplification was observed for most samples, but this did not always translate to high-quality sequencing results. The sequences shown on this slide were obtained from the products shown in lanes 1 and 4 from the left side of the gel. One of these results was very poor quality and showed many base calls with poor Phred scores. The other passed some of our quality assessments. The vast majority of the calls had appropriate Phred scores, and the primary sequences were observed at the end. However, the overall sequence length was a bit short. This was also reflected in both sense and antisense reads. We compared the resulting tiled sequence to references using BLAST, and we found that there was a 33 base pair deletion in the middle of the sequence. This deletion was not supported by any published references. This suggested that we recovered a CO1-like sequence rather than an actual CO1. As a result, this was excluded from further analysis. This example was produced from a pupal exuvia for Blazocypha plintopyga. This observation was not unique to this species or sample type. We found an additional example after testing adult leg taken from Formia regina from long-term storage. This produced a 12 base pair deletion despite the sequences passing the other quality assessments. Throughout our entire validation, we did not observe these deletions from any newly collected specimens. This suggests that these products may be brought out under low template or high degradation circumstances. The next experiment was a test of our internal implementation of the assay. We conducted a blind analysis of specimens that were provided by an outside expert. The purpose of the blind analysis was to show that we may produce accurate results without introducing contamination. We were provided 8 specimens. Some were larvae and some were adults. All 8 were colony bred, so their identifications were known, but these were withheld from us until the experiment was completed. The analyst running the experiment did not have entomology training, so they were not capable of examining specimen morphologies themselves. The PCR products were visualized by gel electrophoresis, which showed clean bands for each specimen. Sequences were obtained for each specimen. These passed the quality assessments described for sequencing data and were tiled to produce the full-length 514 base pair sequence. The sequences were compared to published references using phylogenetic analysis. The references included for this analysis represented populations from the United States. We found that the 8 specimens clustered with one of 4 species groups. 1 clustered with Chrysomya ruffifacies, 3 clustered with Cochlea macellaria, and 2 clustered with Blazocypha plintopyga. These received at least 98% bootstrap support. The final 2 specimens clustered with the Lucilia serracata-like haplotype of Lucilia caprina. These received 90% bootstrap support, which was less than what we observed for the other comparisons due to the similarities between caprina and serracata haplotypes. This type of experiment is very important for our internal quality management. One of the challenges we have discussed is trying to determine how to regulate the proficiency of our personnel for performing these types of experiments. It will likely be through these assays where we will test the ability of an analyst to reproduce data for known specimens. Now I'm going to describe data produced from specimens collected in Harris County, Texas. We expect this data will be helpful for matching unidentified specimens to local populations. Adult flies were caught in baited traps placed in Harris County over the past year and a half. All specimens were identified according to morphologies by a board-certified entomologist. We have sequencing data for more than 100 specimens that correspond to 19 different groups. The vast majority of these specimens are from groups that are relevant to our casework. We performed analyses to understand the differences between and within the 19 groups. This was done using molecular evolutionary analysis software, or MEGA. This slide shows pairwise interspecific differences. The lowest rate for any comparison was 5%, which corresponds to about 25 nucleotides. This was observed between Calliphora vicina and Sinomya cadaverina. Between blowfly species, the rates ranged 5 to 15%, with most instances being greater than 10%. The differences between the flesh fly Blazocypha plintopyga versus any of the blowfly species range 14 to 17%. The differences between the scuttlefly Megacillia scalaris versus the blowflies or Blazocypha plintopyga range 24 to 29%. Intraspecific variations were calculated for prominent species with multiple observations. No variation was observed for several groups, in particular Chrysomya megacephala, Chrysomya ruffifacies, Sinomya cadaverina, and Formia regina. Rates for all other species were less than 1%. These differences amounted to some plurality of individuals having 1 or 2 nucleotide differences. These rates were far less than any of the interspecific variation rates. Because of this, we expect that this data will support identifications when unidentified specimens are consistent with these groups. In supporting these identifications, we would look for specimens that match individuals of these groups or that have only a small number of nucleotide differences. I would like to describe a few additional interesting observations that we made for some groups. This slide shows the portion of a phylogenetic tree comprising the sequences we generated for local Lucilia caprina. We found that most encoded the Serracata-like haplotype. This is known to be widespread in the United States, so this was the natural expectation. The local specimens and published references did harbor polymorphisms, and this caused the bootstrap support to be reduced to 68% for the group encompassing all of the sequences, but all of them contained a core group of 4 polymorphisms that distinguished them from Serracata. We also observed one specimen that clustered more closely with the so-called pure Lucilia caprina references. It still did not match very closely with any of the references that were included in this analysis. This is likely a transparent population that has its own differences. It will be part of our future work to find more specimens from our area that match this specimen. We also identified a number of Lucilia exemia. Each of these showed the Ceruliviridae-like haplotype. These clustered with published references for exemia as well as Lucilia mexicana and Ceruliviridae from central and eastern parts of the country that share this haplotype. We did not detect either Lucilia mexicana or Ceruliviridae among our adults in the current survey. We also did not recover any unique regional haplotypes for Lucilia exemia. There were a number of additional species that were observed in our survey. These included flesh flies, which so far we have been unable to verify by morphology alone. DNA analysis suggests that these are members of the genera Sarcophaga, Oxysarcodesia, Ravinia, and Helicobia. One of these was Sarcophaga hemorrhoidalis, which is relevant to casework in other parts of the United States. We have also observed other associated species, Musca domestica, P. affleckasii, and members of genera Hydrotia and Phania. Many of these specimens are described here to the genus level. This is because the results are based on DNA analysis only. We are in the process of having these adults examined by other experts so we can confirm their identities to the species level. At this point, I have described our assay, which is useful for identifying specimens belonging to various insect species that are relevant to forensic casework. I have also described quality control measures that may be applied and several validation studies that support implementation of this assay. These include evaluation for sensitivity, challenging reaction conditions, blinded experiments, and statistics for local populations. For the final portion of this presentation, I'm going to walk through an experiment for analyzing unidentified immature specimens. We support their identifications using comparisons to published and local references. We tested 17 specimens. These were larvae, pupa, and pupal exuvia that we were not able to identify by examining morphologies. They were collected during casework that occurred from 2014 to 2017 and held in long-term storage. CO1 amplification was tested for each specimen, and products were visualized by gel electrophoresis. The appropriate band was observed for all larvae and pupa that were tested. This was observed for only one of the 7 pupal exuvia that were tested. This lane is shown in the top gel using an asterisk. The remaining exuvia did not yield the appropriate amplification product. Most did not show any amplification. One showed a second band at around 500 base pairs, shown here using the arrow in the bottom gel. And the last specimen produced a single band that appeared to be somewhat smaller than the expected size, which is shown in the far right of the bottom gel. The DNA extracts from the larva and the pupa were very high relative to the amount that we needed for analysis. These sample types are quite fleshy, so it is easy to get way more DNA than you need. However, the pupal exuvia typically do not yield very much DNA. We would process the entire exuvia specimen, but this did not seem to improve outcomes. However, some pupal exuvia contained a small amount of residual tissue, and this may have improved outcomes. Sequencing was performed for the remaining 11 specimens. These were the 2 pupa, 8 larvae, and 1 pupal exuvia. All sequences passed the quality assessments made for sequencing data as shown here as an example. The majority of base calls showed appropriate Phred scores. The primer sequences were observed at the end of the sense and antisense reads as shown underlined. And the tiled sequences were the expected length. The full-length 514 base pair sequence was obtained for each specimen. Statistical analysis was performed using multiple methods. The first was through phylogenetic analysis. This slide shows excerpts from the tree where the casework specimens fell. The analysis included published references and sequences observed from local specimens. The majority of the specimens were supported as Blazocypha plantopyga. 2 others were supported as Formia regina, 1 as Chrysomya megacephala, and 2 as Calliphora vicina. At least 96% bootstrap support was achieved for all clusters. These included analysis for the 1-pupil exuvia, which clustered as Calliphora vicina. Another analysis that was conducted was through BLAST. Queried specimens were compared to published references in GenBank. On the left is an analysis for a putative Formia regina, and on the right is a putative Blasecypha pentapyga. In both cases, there are high scores for coverage and sequence identity for appropriate references. Natural breaks may be observed for both the BLAST scores and percent identities, and these occurred where matched references were no longer representing Formia regina or Blasecypha pentapyga. In the future, we expect to enter sequences from our local specimens into GenBank. This shows an example BLAST comparison between an unidentified specimen and a verified local specimen. Nucleotide differences are circled in red. There are only 1 or 2 differences between these unidentified specimens and the local references used here. Similar results were observed for each of the unidentified specimens. Differences of this magnitude are consistent with the variations we found in our population study. An additional type of analysis that could be used to support identifications would be through the MEGA software, which we previously used to help describe our local populations. We combine sequences from unidentified specimens with those we obtain from populations in Harris County. We then use the software to calculate the average differences between unidentified specimens and the verified local specimens. One specimen was proposed as Chrysomya megacephala. No difference was observed between this specimen and the locals. The remaining specimens showed rates ranging 0.06% to 0.61%. For some of these comparisons, the unidentified specimens were identical to some individuals, but different from others. In all cases, the rates were less than 1% and similar to the interspecific variations observed for local populations. This slide shows a summary of this analysis. Identifications were supported using phylogenetic analysis, where clustering was observed with 96% or greater statistical support. These were also compared to verified specimens originating from Harris County. Most of the unidentified specimens produced a sequence that was identical to at least 1 of our verified locals. The remaining 4 showed 1 or 2 nucleotide differences. In summary, we are validating a method for taxonomic identification of forensically relevant insects through CO1 barcoding. The method is effective for specimens which may not be identified to the species level by morphology alone, such as those lacking distinct morphological traits or specimens that are damaged or poorly preserved. We are simplifying this process by describing a single primer set for amplifying species that are relevant to forensic casework in Southeast Texas. Our list includes members of various blowfly genera, as well as the flesh fly Blasecypha pentapyga and the scuttlefly Megacilea scalaris. We demonstrate methods for supporting identification. These are phylogenetic analysis and BLAST, which allow comparisons to published references and verified local specimens. We additionally describe the use of MEGA software for making comparisons within and between populations. As part of this presentation, we have described multiple characteristics of our assay. We have demonstrated analysis of a variety of forensically relevant species and tested the assay under a number of adverse conditions. We conducted a survey of local populations, which we have used to build expectations that support the analysis. And finally, we demonstrated the assay for unidentified specimens from our past casework. In the future, we are working towards getting into active casework. We're preparing our data for peer review. Specimens used in our validation will be submitted to an entomology museum. These will include all of the adults collected in Harris County and the unidentified larvae and pupa that were tested during validation. We will also be submitting sequences to GenBank for any adult flies whose identities were verified by an expert using morphologies. We're also in the late stages of our validation and preparing documentation for internal reviews. The documentation will include formal summaries for each validation experiment, detailed standard operating procedures, and recommendations for analyst training. We hope to have much of this initiated over the next month and be working casework sometime in the new year. Our future research goals would include additional surveying of species inhabiting our area. We expect to continue surveying adult specimens that may be incorporated into our analyses for local populations and compile new data describing species encounters in casework. We hope to produce sequences for species that presently have little published information and build data for additional species. I would like to thank our team with the Harris County Institute of Forensic Sciences, including Dr. Michelle Sanford, Michael Donnelly, Katie Welch, and Dr. Roger Kahn. We would like to thank Dr. Jeff Tomberlin, Dr. Terry Whitworth, and Dr. Greg Daglin, who have donated specimens and assisted with identifications. We would also like to acknowledge support from the NIJ and the Forensic Technology Center of Excellence for allowing us the opportunity to present our work today. Thank you.
All right. Thank you, Dr. Kwiatkowski, for such a fantastic and informative presentation. Let's go ahead and get into some questions from the audience. First one I have here, for species with overlapping geographical distributions, do they have similar TOC, or would a species misclassification lead to large errors in the estimation of PMI? And they also noted they're referring specifically to the ones that also have similar phylogeny.
Yes. So in this group, there were two groups that we were concerned with in this study. And so really the answer depends on which group you're talking about. It's going to be different for each one. So one of these groups was for between Lucilia serracata and caprina. Now, between those two species, there are indications in the literature that the development is different enough that it would lead to an error. Now, that itself, wouldn't necessarily defeat the use of time of colonization, because even though you could still be wrong in defining a range of times that could lead to PMI, it would still be possible to report an absolute minimum, which in this case would be through Lucilia caprina. And so there are situations where you could, if you needed to, use that data set to define just an absolute minimum, because that information, even if it is incorrect, could still be useful in some situations. For the other group, that was the group with Exemia and Cerula viridis and Mexicana. So that's a different situation because we don't have development data for all of those species. So there is some data for Exemia. There is not data for Cerula viridis. And actually for Cerula viridis, it's notorious for being difficult to rear in laboratory conditions. So we simply don't know the answer if there would be an error or not.
Awesome, thank you. Next question I have here, have you considered using additional primer sets to identify hybrids of closely related species?
So yes, it's something that we've considered to try to help us with that problem, but it does have its own challenges. So there is certainly precedent in the literature for using other markers, nuclear markers in particular, for being able to distinguish between the hybrids. And it's definitely possible to pick out hybrids from the population. But the problem is that these species that are hybridizing, it's the possibility that they're engaging in modern day hybridization, something that is ongoing. And so, for that group, we can't always expect that these markers are going to be fixed in those populations. So the question then becomes, well, not only what markers, but how many should we use? Because the more markers you use, the less chance you're going to have of missing them. And so what we really need to do is to be able to do some type of comprehensive study of these hybrid populations in the area and see, well, what are the markers that might be useful? How many would we need in which ones to catch the majority of the species here, and be able to really reasonably separate the parent species from those hybrids. So it's definitely something that is possible. It's something we're thinking about, but we just have some questions and things that we'd like to work through with it first.
Absolutely, thank you. And just wanted to go ahead and remind the audience, we've got plenty of time. We've got a couple more questions in the queue, but if you have anything else that you'd like to have Dr. Kwiatkowski answer, please send them into the chat. We'll make sure we have time to try and answer everybody's questions. Next question I have here is, what do you do with partial sequences? Is the sequence length the same for all species?
So when we're doing our analysis, we look for the full length sequences. We haven't had any problem with getting those full length sequences at all. And so for all the sequences that are in the study, all of those are the full length. Now and so that length it's 514 base pairs for this primer set when you take out the primers on the ends. There actually is one exception, which was for Megacelia scalaris, which has an extra codon right in front of the stock codon, and so that's 517. And so that's something that we've observed in our work, and also that we've seen in published sources as well.
Another question here. In wildlife forensics, we routinely ID species of animals and plants in trade. We have not been able to produce a statistic indicating surety, bootstrap give node support, not surety. Have you?
Right. So the question of statistics and what's the best statistics to use, this is something that we're trying to work through ourselves. So what we really need is we need some form of statistic, whether it's node support through the bootstraps or surety, that type of thing. What we just need is some type of mathematical something that will help us to separate. For the species that we're looking at, we're really not trying to separate the ones that are really closely related at all. We're looking at the ones that are very different. So even something like the bootstrap support, we think is going to be sufficient for just giving us some kind of support for those differences. Now, if we did try to distinguish between those that are much more similar, then we might need to have a more robust measurement for sure.
Gotcha. Next question I hear: for similar phylogeny specimens that cannot be accurately differentiated with this method, would it be worthwhile to do a follow-up analysis with primers targeted to those species?
Absolutely. And so it definitely would be something that we could do. One of the things that we would want to think about is in terms of these questions of these different, because there are multiple groups that we have the same problem with. And so what would really be great is if we could identify primers that would work with all of those groups. That way that would help minimize the amount of work that we do. Definitely a point of what we're trying to do is to minimize the amount of work that we're doing, because we are a forensic genetics crime lab. We don't have a lot of resources to put towards this type of analysis. So anything that we can do to minimize the amount of work we do is fantastic. But it definitely would be useful to think of primer sets that are targeting those species, and maybe some plurality of these groups that would be useful for that purpose. Yes.
Awesome. Well, want to put out just another call for any other questions. We are getting towards the top of our queue. And while we're waiting on those questions, Dr. Kwiatkowski, do you have anything that you want to reiterate? Anything that you want to bring up again? Just anything. The floor is yours to go ahead and provide any more details that you'd like.
No, there's nothing off the top of my head that I'd like to add to the work, but I would just like to reiterate just thank you to the Forensic Technology Center of Excellence for allowing me to give this presentation, and also thank you to the audience for watching and for these insightful questions. Thank you very much.
Awesome. Well, thank you, and we appreciate you having here. And I do want to ask and see if you have any more details on. You put the slide at the end on the future of this project and this study. Any other details on what you're looking to do, what you're hoping to do next, and maybe even beyond just this particular study? Is there any insights, anything we should keep an eye out for down the road?
It's something that we're at a bit of a stopping point with the current project, and so that's very much part of our ongoing discussions of where are we going with this next. Certainly, what we're trying to do right now is to get this going for casework, so that we can start to actually get this into motion and get this into the lab, so that we can start using it. As part of that, what we'll look to do is to continuing surveys of our area to try to continue to build on the database of local sequences that we have. We were able to get representative specimens for just for most of the species that are relevant for our area. There are still some that are less common that we didn't pick up on our first go at it. So that's something that we'll always be looking for to boost our work. The other work that we are thinking about does follow along the lines that we've seen in the questions, which is what can we do to try to do some more explorations with these that are closely related. And so that will be just thinking about, can we do something with regional populations? Can we see if there's some way that at least within our region that we can differentiate these with a specific marker somehow. Is there something that we can do to further expand this work? Other things that we'll be looking into will be trying to expand a little bit more out into flesh flies, of course, because we didn't have too much that we're really focusing on with this particular study with our area, but there's certainly other species that are going to be helpful for other areas around the country. I think a big goal of this work is to build something that other laboratories would be able to replicate. And so being able to expand the species that we're able to work with is something that would help us a long way with that.
Absolutely. Awesome. Well, I think that is the end of all our questions. We didn't have any others come up. So we'll go ahead and start closing things out, and start off by just, again, saying thank you to you and for taking the time out of your day to present the material for today's webinar and all the expertise and answering those questions. So Dr. Kwiatkowski, thank you again for taking that time to provide such a wonderful presentation.
Sure. Thank you.
Before you leave today's webinar, we ask if you could participate in a short 5-minute survey. You will see a checkbox icon directly above the chat. Simply click that button to be linked to the audience feedback survey for today's event. Your feedback about our webinars is critical information that helps us improve and continue to provide you the best webinar experience possible. If you could please take those few moments to complete the survey, we would greatly appreciate it. Lastly, across the top of the live stream window are links to all our social media profiles, including Twitter, LinkedIn, and Facebook, a link to our website at forensiccoe.org, and a link to listen to episodes of the FTCOE's podcast, Just Science. There's also a link to sign up for the FTCOE newsletter so you can stay up to date with everything that FTCOE is doing in support of the forensic community. Thank you again for attending, and we hope everyone has a great day.